Hello everyone, welcome to the IBH Seminar. If you would not like your face to be shown, please turn off your camera. This meeting will be recorded and available on the website afterwards. So it's my pleasure to welcome you all and introduce today's speaker, who's Dr. Amanda Souza Câmara. She's a biophysicist who completed her PhD at the University of São Paulo in computational modeling. She did her postdoc in Germany at the Institute of Plant Genetics and Crop Plant Research. And she's entered the world of plant genetics and genomics, and she's currently working in modeling chromosomes compaction mechanisms. And she's going to talk to us today about her work in barley chromatin structural organization.
Okay, well, so that was me. Thank you, Jessica, for the introduction. Thank you, the organizers, for giving me the opportunity to show my work. Just to be on the safe side, because we're having issues before, is everything okay with my presentation? Do you see with this presentation mode? And can I jump to the next slide? You see?
It looks beautiful.
Okay, well, many thanks. I don't see any of you. I don't know if you could just open your mics and say if something goes wrong. Please do. Okay, then I'll start my presentation on barley chromatin structural organization during mitosis. And I'm sure we all here together by our passion for barley, so I guess it doesn't need introduction on why I'm working on this species. But we are working with barley, and we have a great opportunity to work with a general hypothesis. So we had these cutting edge techniques and some starting with high-C results, and we had this opportunity to work on a general coiling mechanism, coiling organization of chromosomes and mitosis using barley as a model. And mostly because we have the genome reference from our own group, and barley has only few chromosomes, 7 chromosomes, and they are mostly large, so it's easy to see in the microscope. And yeah, so we had these nice tools to investigate barley conformation during barley chromosomes, conformation during mitosis. And it's a question that it's been already on for more than 100 years. And at the beginning, when they first observed chromosomes under the microscope, it was clearly that it was a helix. So it started with Baranetsky observing these chromosomes in Tradescantia. But there was early experiments, and as the years go by, then some criticism rise about the way that they were observed, and maybe artifacts from the technique. But as the years go on, some other models appeared, but helical conformation was always inside of our minds or in the back of our head. And different kinds of models, but still with a helix were proposed. And most recently, there is this butterbrush model proposed by high-C data. This was with chicken. Well, anyway, after and until the discovery of the DNA double helix structure, it kind of makes sense imagining that the DNA would further coil into levels of helix. But even this, it seems very hard for the chromosome to be a helix in all levels of organization. And other models were proposed, many other kinds. So we try to say helical models and non-helical models. And inside the non-helical models, there's a huge diversity on how they can be, and the techniques they use to propose them. So radial loops, some matrices, a network of chromatin with different proteins, layers, and even more recently, there's after the last one proposed from high-C that should be a helix. Then there was this other non or half helical model still with microscopy. And well, so that's the topic that we wanted to address, having these nice results that we find out. And now starting explaining maybe the high-C technique that was our starting point in this research. And maybe it's already very common technique for you, but I'll quickly explain because it's important to understand how we infer from this that it's a helix. So in the high-C experiment, the first step is to fix chromosomes with formaldehyde. And then the many pieces of the chromosome are cut with the digestion enzymes. And then these pieces are religated. So it might happen that pieces of chromosome that were not close in the linear sequence, but are close in space, that they are ligated together because of close spatial proximity. And then these pieces are sequenced, and then we have a library, and this one we can map onto the genome reference, and we see which pieces of the chromosome are close to each other. And in the end, we have a matrix like this. So we have this main diagonal, which indicates that pieces close to each other and in the sequence are also close in space. But some parts that are not close in sequence can also be close in space. So this is the main technique, so-called chromosome conformation capture. And later was developed the high-C, which is for the whole genome. And high-C was used by our group to assemble the genome for barley. So early in 2017, it helped the genome assembly because when the genome is wrongly assembled, then it's easy to see in the matrix that there will be some parts inverted and gaps and strong, very discrepant signals around the diagonal. But this would be a very clean high-C matrix, but for interface. And already in interface, we can see, apart from helping the genome assemble, well, the main purpose is to investigate the conformation of chromosomes. And here in interface, this pattern with this anti-diagonal, it indicates this rubble conformation. So when chromosomes are resemble their anaphase stage, and they are pulled out towards the daughter cells, and you can see each arm side to each other, and centromeres in one side and telomeres in the other. And this is clearly different from humans. So in humans, we see this kind of squares and tabs and forming domains and compartments. And so there are many differences in barley and humans or animals in general, from plants and animals. And but we decided to go beyond interface and check metaphase as well. And to do this, we were collaborating with the group of Hanna Sinkova and the Institute of Experimental Botany in the Czech Republic. And they performed cell cycle synchronization and flow sorting. This was the job of Petri. And yeah, they used root cells that are fast dividing and flow sorted mitotic chromosomes, and then started the normal high-C protocol. So barley was one of the first organisms, one of the first species to have high-C data for mitotic chromosomes. It was just behind chicken. It was published in chicken's chromosomes. It was published in 2018, and axolotl chromosome published in 2021. But they also showed this very similar pattern, which is second diagonal parallel to the first one. And it's different from interface that we saw an anti-diagonal only. So even between comparing very different species, there is this main pattern that's the same, and makes sense because all of them need to go through this compaction process to reach mitosis, and maybe they have the same process. So an interesting thing to investigate. And the second diagonal could only translate into a helical organization, as far as we know. There haven't been proposed any other model that could explain this pattern. And this is so because, well, and the first main diagonal, we already know that's because close sites close to each other in the linear sequence, they are also close in space. But we see that distant sites sometimes are distant in space too. But if you come back an entire period, then you have, again, the distant sites are closer to each other. And this happens periodically along the entire chromosome. So we see that there is a pattern that it's always this distance between sites that is distant, it's separated by this number of bases, and they always come together again in this helical pattern. So that's why we see a wider space between the two diagonals and then a strong signal again. And this was in the work with chicken. They proposed that it would be called bottle brush model. That they proposed this with polymer modeling. That there should be a helical scaffold of proteins, and from that, chromatin loops emerging out of it. There is another way of analyzing the Ising matrix, which makes it easier to compare between species and between chromosomes, or to make comparisons. And it's the analysis of the contact probability. So imagine that we take orthogonal line here from the center, so from this diagonal, and we just check for its intensity. We would see a pattern like this. So if you take the average, so along all the chromosome, we see that high intensity at the beginning, corresponding to the strong signal at the diagonal, and then it decays. And this is where we see a lighter signal in between diagonals, and then we call this a bump. That's where sites come back close to each other again. And this is what will be the corresponding to a helical turn. So at this distance, then we will always have sites of the chromosome coming back together. And this relates to the helical turn length. And for barley, it has 7 chromosomes, and all of them showing a bump at the same place, although they have slightly different chromosome sites. And well, with the contact probability, it's easy to compare with other species. So comparing barley and chicken, we see the same pattern of this bump. But for chicken, well, here in orange, it's the bump appears much earlier than for barley. And the same comparing axolotl and chicken. So axolotl again has a bump at larger distances, and it's summarized here. So chicken will have in this model a helical turn of 8 megabases, axolotl 35 megabases, and barley 30, but all indicating a helical organization. With this contact probability, we decided to went further and not take just the average, but look into the local contact probability. So we take along the chromosome regions of 5 megabases, and we calculate the contact probability again and see where the bump is. And we can see in this movie that the bump changes according to the place of the chromosome that we are analyzing. And it's interesting to see that it matches around telomeres and around the north and centromeres that there is no signal for helical organization, which could be a sign of a different organization for these regions. So this will be the result of this analysis of local contact probability along the chromosome and for chromosomes of barley. So if we can see that centromeres and this north, so this second constriction, and it's not indicating a helix. And we can see that it's kind of stable in the chromosome arm, but it decreases fast in the telomeres. And especially for chromosome number 5H, there is this kind of different decrease in the helical turn length, and it matches the amount of genes that we found in this chromosome. So here in this graphic, we show the bond position, which relates to the helical turn size, and the gene density. So we found that this pattern of the helical turn correlates with gene density. But how we propose, I'll show to you how we relate these features and for after. So HiC, it's the most recent technique used to analyze chromosome conformation, and it's we're very confident of its results, but there are some criticism. So imagine that we have to have in mind that HiC uses a population of cells, and we might be detecting chromosomes in different conformations. Now, although we have it always in mitosis, we don't know how plastic they are, so they can be a bit different. And if we consider that they have as many conformations as like us, that we could move and we change our arms and legs, and we can adopt different conformations. If we take the average of this, we will have a person that looks more like a butterfly than a person. And this is simply not the case. So we must be very careful when taking the conclusions on the average. And to help us with that, we came back to microscopy. So we designed oligo-FISH probes to try to check individual chromosomes and if we could see this helical conformation in individual chromosomes. So this experimental part was done by Ivona Kubalova in collaboration with the group of Andreas Hu here in IPK too. So they took this region of the chromosome 5H, which I told you that was rich in genes. So it has many unique sequences that was perfect to design these oligo probes. So we designed it like this. We try to, having in mind that we know the helical turn for barley, that was around 30. We designed so we could have 3 full turns that we would see. And in the end, how telomeres, have some idea of how telomeres can be formed. And at the beginning, 2 half turns. And this is how we observe the chromosomes with this painting under the microscope or using super resolution microscope. So these are chromosome 5H, easy to recognize among the others. And we clearly see these 3 turns of the helical organization, one above the other. And it's what we expect from the helical organization. So you see the turns one above the other like layers. And the most interesting is to see the half turns side by side. So we could see here really a half helical turn and the other one following it. But it was not always the case, as sometimes the turn can be like half turn can be in front of the other. So we would see here just the white one, and behind it we have the green one. And well, not to just stay in this image observation, we try to quantify the helical turns with this technique as well. So just to have a notion of where these oligoprobes are in the position in the chromosome, regarding the helical train that we calculated from HiC. So this is the helical train length, and each region what helical train length we expect. And it's funny to see that when you measure the height of of each pattern that we observe here, so this height of each turn, it follows this pattern of the turn size that we expect. So yes, so turns that are longer, we expect to see just the size of one turn, and regions that we have smaller turns, then we expect this region to cover more turns, and hence the height of the turn of the band that we observe in the chromosome is also higher. And it's funny to see that right in this region for probe that we called MOA, there is this increase in the helical turn, and we see the decrease in the height, or at least not an increase as would be the case of a linear decay. So this was measuring the height, and we decided to measure also the volume of the probes. So we see here that half helical probes have smaller volume, the entire turns probes, and these black dots indicating us the percentage of amount of DNA that these turns cover. So we expect 5.7%, for example, for this probe, so 5% volume. But we always observed volumes, we always measured volumes a bit smaller than we expected. So it might be something about the way we're measuring it. But it's interesting to see the flamingo, the last probe in the telomeres, it happens the opposite. So it's the only one that has a volume even higher than expected. So it's more likely that the telomeres are less dense compared to the rest of the chromosome, compared to the arm. And well, microscopy could give us further support in a different kind of experiment, which is called cystic chromatid exchange. And it forms this Harlequin pattern. So before duplication, so we put these different probes to color the duplicated chromatid. And we can see that there is this exchange of chromatids between the sister chromatids that it shows as a Harlequin pattern. And these segments of sister chromatid exchange, they also look like fragments of helical turns. So we decided to quantify them. And we are always looking for segments as small as possible. And we saw a pattern that when they are very small, they always had around 400 nanometers in height, but didn't cover yet a full width of the chromosome arm. But when it covers the full chromatid width, then it would only grow in the height direction. So we would never see a sister chromatid exchange like this one that's very tall along the chromosome arm, but it's very thin. And this kind of segments, they look like segments of the helical turns too. So this one would correspond to small turns of pieces of a turn, and then a full turn, and then only full numbers of turn and a bit more because the chromatid width is always covered. So this was another way of supporting the helical organization. That it's very interesting because it doesn't need a genome reference. So maybe it's easy to try in other species that still don't have a genome assembled. And okay, so we were very confident after high-C data and microscopy indicating a helical conformation, and we wondered how is this helix formed. So the work done with Chicken, in that work they proposed that this helix should be formed by a protein scaffold that's made of condensins 1 and 2. And so, and from this scaffold of proteins, loops of chromatins would go out and would emerge of it. And depending on the stage of condensation, of compaction, this scaffold forms a helix that has smaller or bigger turns. So they detected this bump moving with the compaction. So this is late prometaphase, and this is early prometaphase, prophase. And condensing 2 will have a very central role in this model because they did the depletion of condensing 2 or condensing 1, and they noted that when they depleted condensing 1, there appears this pattern in the high C that the bumps is still there, but not this region. And when they depleted condensing 2, then the bump wasn't there anymore, but this lower level was still correct. So they proposed that condensing 2 would form the helical scaffold, and condensing 1 would come side to it, forming smaller loops of chromatin. Condensins are part of this group of structural maintenance of chromosome complex proteins, and they can work as loop extruders. So they have this shape of a ring, and they bind unspecifically to DNA or to chromatin, and they move like they were extruding a loop, like in this nice represent movie. And and if you put a few of them, they form these loops, and it's supposed that during interphase, this type of proteins is forming these loops to connect distant regulatory sites. So they bring distant sites together and form these regions where there should be regulation of genes and different genes, the regulation of different genes. But if you put many of these type of protein, then they start to compact the chromosome, because they also serve as limiting, so to other condensing. So as they extrude the loop and find another protein and another condensing extrude in the loop of chromatin, they stop and they remain side by side, and from there emerges many loops. So depending on the amount of condensins that you put, or that there is in the chromosome, then it's more or less compact. So I tried to observe this with a polymer simulation. I'll show here is the movie, but please don't mind this red bits. They were just randomly selected, but they do not interfere with the loop extrusion process. So we see an initial conformation that resembles interphase, and now we have these loop extruders extruding loops. And it works like this. It binds anywhere, and it moves as to extrude this loop, to leave this chromatin loop behind, but it also can dynamically unbind from chromatin and bind somewhere else. So as these condensings are very dynamic, although they are very dynamic, they form this very stable condensed state. And we can see that it forms this condensed state, but it forms the condensings. Sometimes there are clusters of them, but they are always side by side. So we can see that they forms like a scaffold along the chromosome that is compacted by loops. So it makes sense, the model that they proposed, that maybe these condensates really do from this scaffold, that in later stages of compaction, they would make a helical organization. And there is further evidence that parts of these condensings went in the crystal. So in experiments of crystallography that they did to solve the structure, they see that they form some kind of a helical conformation, this part of the condensing. So maybe they could assemble like this when they are into many. So we took this model. Assuming this model, you can then take further parameters from the high C contact probability. So you can adjust a model where the contact probability would fit the experimental contact probability. So in this way, you can just build these polymer models, calculate the contact probability for them, and compare with experimental data. And the best ones, so the ones that would fit the contact probability, should then be likely to be the conformation of the chromosome. So this is the approach that they did, Giptos et al. In the work with chicken. And we decided to do the same for barley. And I tried this way more than 800 models, varying terrain and major and minor loop sizes, but it was never quite the same as in the experiment, and especially in this region. So we didn't seem to find the sizes of the loops. And there's actually a very simple reason. So we came back to the contact probability of metaphase of barley to see if we have any clue. And he actually came from the group of Hannah. They suggested looking at different analysis of the contact probability. So here we take the derivative, which is easier to see this kind of plateaus. So it's closer to, when it's 0, it means that it's either this value or this top. And when it's closer to it, then it's more likely this plateau. So we can see that there is this increase in the derivative at 500 kp, that for interface there wasn't. And just taking the difference between the metaphase and the interface contact probabilities, we see an increase at 3 megabases too, apart from the 30 megabases for the helical turn. So this analysis really seems to indicate that the helical turn will be indeed 30 megabases around it. But for the regions of lower level organization, then we see this kind of pattern rising around 3 megabases and 500 kb. So it might indicate the sizes of the loops. So we tried this one, but the model parameters wouldn't match, simply don't match for barley. And there is a simple mathematical reason for this. So I hope you can permit me to fill this table. And we start with this simple assumption. The chromatin loops are always side by side, and these condensing complexes too. So they form this scaffold and they need to be very close to each other. So we set this, the model proposed for chicken. They had this distance set between these bases of the loops, which is just 2 times the size of a nucleosome. So you can imagine here that the condensing would bind 2 nucleosomes together, each one being 10 nanometers. So 2 nucleosomes together will be 20 nanometers. And because the loops are side by side, then the next base will be very close to it. So we have always two nucleosomes here, two others here, already being the basis of the loops. So this will be the distance between condensing two complex, 2 nanometers. And we assume the same for barley because it's a very conserved protein family. And well, then it changed a bit each model for each species. So the turn length of chicken was 8 megabases, and the turn length for barley was 30 megabases. And from our analysis, the major loops should be 3 megabases compared to chicken that was only 400 kb. And the turn height we had from microscopy should be 400 nanometers for barley. And for them, they had in the model 200 nanometers. Okay, so if we have each turn of 8 megabases for chicken and major loops of 400 kb, it means that they have 20 loops of chromatin per turn. And in the case of barley, this should be only 10 chromatin loops per turn. So if we have these loops and they're always side by side, then, well, 20 times 20 nanometers for chicken, we should have a turn length that's around 400 nanometers. So this helical path here is 400 nanometers. And it should fit inside a 200 nanometers helical turn height. So this is good that this turn height is like the limit, and the helical turn path should be inside it and should be bigger because it needs to coil, so it cannot be straight. But for barley happens the opposite. So we have a turn height that's 400 nanometers and a helical path that can only cover 200 nanometers. So it simply didn't match, filling the helical turn with condensings side by side. So we thought, well, maybe they're not side by side. And we suggested that these condensings would be a bit farther apart from each other. 20 nanometers is the distance between each nucleosome bases forming the loops, but then 4 nanometers should be between each condensing complex. And we tried different distances, and we see that the minimum would be to make a linear scaffold. And if when condensings are even more apart, then they form this helical path. And well, it then fitted the contact probability within some error. So we see that condensings are no longer forming a continuous helical path, but they are clustering along the chromosome axis. And well, we further took this analysis to see if the loop, the length of the loop, have something to do with this spacing. And it turns out that there is. So the bigger the loop, the more distance this condensing complex needs to be from each other. And so we wanted to check in back microscopy or supporter if this will be true for chromosomes. And indeed, when we try monolocalization of condensate, we don't see a helical scaffold, but these clusters. And well, other experiments support this. And also transmission electron microscopy, it showed this kind of stacks in the size of 100 nanometers. And so maybe there is, then there's no protein scaffold with evidences from other experiments. So just making a summary for barley, you see this amount of turns that are variable along the chromatin, the arm, and the turn should be 400 nanometers, and these loops are the kind of intermingle. So we don't clearly see separated turns in the chromosome. So this was our conclusion from this study. But if you allow me some speculations, we then turn to see what makes the chromatids coil. And well, there is the formation of the loops is still hold, and maybe there are some cases where this kind of structure, which we then named the chromonema, wouldn't need a protein scaffold, but it, well, different mechanisms can yield a chromonema with no protein scaffold. And well, remembering it's made of chromatin loops, it's a bit hard to know how they could interact with each other, because these loops can repel each other when they're too close. So it might be complex mechanisms to bring them together. But if I can bet, I would bet on entropy. Well, after the nucleus membrane is broken, then it's already proposed that this kind of ropes would coil in the presence of a different solvent that causes these excluded volumes to come together. And there are the proteins doing the coating of the chromosome, so they can be protected from the cytoplasm when the nucleus membrane break. And well, the coil of ropes has been studied before, and they noticed that there are some helical organizations that are possible and others they are not. So my future plans is to compare this possible organization depending on the length of the chromonema, so the radius of the chromonema, and to see if there is really some constrictions on the size of the chromosomes. And the idea is that for the same species, the loop sizes should be always the same regardless of the size of the chromosome, because it depends only on the concentration. And so if you have longer chromosomes, you will have always the same loop size. And longer chromosomes could then coil, but smaller ones could not. And again, with chicken, if you're not tired of chicken yet, we can see here that regardless of the size of the chromosome, it's always a turn in the same position, but smaller chromosomes don't even come to coil. And so just comparing in this chromonema coiling mechanism that we speculate to be, it would be proportional to the genome size. So bigger genomes form bigger chromosomes with bigger turns. They are inside bigger loops. So I'd like to thank you, these people that participated in the project, Ivana, who did the microscopy experiments, and the group of Hannah and Petri, who did the high-C experiment and the flow sorting. And well, I'd like to thank especially Fight for discussing with me the chromonema and the helical models. And well, Martin Marshall, my supervisor, who called me to participate in this project, well, who allowed me. And he's been very supportive with my career, and Hannah as well. So thank you, the organizers, for giving me the opportunity to present, and thank you for your attention. Many thanks. I can take questions if you have any now. No.
Amanda, there are no questions in the chat section. It would just be if anyone wanted to raise their hand and speak up with a question for you.
Well, I can see a question in the chat. Maybe I can read and try to answer.
Oh, that's just popped in. Good, thank you.
Well, the question is from Sebastian. He says, very interesting results. Many things. Have you looked into possible correlations between condensing family expansion and contraction and the influence on loop size? Family expansion and contraction, I imagine between different species. But no, I didn't see, but it differs a lot. So for example, in animals, when it's been seen that condensins 1 only enter after the nucleus membrane breakage, but it seems that in barley and Arabidopsis, they are already there in interface. So I'm sure there are differences of how they work in different species, and I would love to check in the loop sizes in different ones. Let me know if it answered your question or if you have further doubts. Okay. Well, I hope you have enjoyed anyway, and I'm always open to contact. If you want to reach me, you can find easily in IPK my email. Oh, I can also leave it here in the chat if you want to reach me afterwards. I would love to have your feedback on the coiling mechanism that we are proposing.
I think most of us are probably just a little bit blown away. I've not really seen very many presentations like that, and it's really kind of interesting and fascinating, I guess. Well done.
Well, many thanks.
I think we'll just finish there then, Amanda, if that's okay with you.
Sure, yes. Well,
great. Thank you very much, and thanks
Yeah.
everybody for joining us.
Very interesting, Amanda. Thank you. Thank you.
Thank you.